Hi, assalamu alaikum, and a good day to everyone. Thank you for tuning in to my presentation. Today, I will present to you my extended essay research in biology subject. My name is Wan Agdim Hizumi bin Wan Azman, and my EE advisor is Ms. Nohaidi Ismail. My research question is, are the common commercial seafoods, which are blood cockles, squids, shrimps, clams, and mussels in Malaysia, contaminated with microplastic by using gravimetric analysis method? And what are the possible sources of this microplastic contamination in Malaysian aquaculture methods that is considered a major health concern? Now, without further ado, let's move on to the next slide. Generally, to understand what is microplastics, you need to understand, you need to be introduced with its definition. Microplastics are generally plastic granules size less than 5 millimeter caused by single-use plastics and improper waste management. It can be derived from larger plastics through physical or chemical degradation, with household laundry washing contributing the most pollution, or purposefully made for global consumption in skincare products, for example. Microplastics has been shown to negatively impact human health either chemically, physically, or microbiologically. Chemically, it can leach bisphenol A or BPA. It can cause endocrine disruption because of its similarity to estrogen, and it can cause low sperm count in males and early puberty in females, and also cause obesity-related issues. Physically, it can cause obstruction in our gastrointestinal tract and our digestive tract. Essentially, when it clumps and it just affects our digestive system process to be able to absorb food and our process of digesting food later on. Microbiologically, it can affect our gut microbiota, or also known as the microorganisms in our gut themselves. These microplastics can deter their microorganism growth, and this can essentially introduce other bacteria and other microbes to grow in our gut, which is something that we don't want. And this is caused by biomagnification of microplastics, which affects the food web, especially humans, as the topmost trophic level. Biomagnification is related to bioaccumulation, where in each subsequent trophic level, the amount of microplastic ingestion increases as per amount of prior organisms that we ingest, therefore causing biomagnification of harms that can affect our system, our body system. And I will be focusing on marine animals, as they are the most prevalent in microplastic pollution, as microplastics are commonly waterborne and then consumed by humans. And we should understand that in the future, plastic demand is expected to grow expeditiously, that can contribute to more microplastic pollution, and is not currently seen as dangerous, as researchers has compared microplastics to cigarettes, for example, as the effects are long term and not direct. And I will be focusing on aquaculture, because aquaculture is a common method of seafood rearing in Malaysia for local and global seafood consumption. Therefore, the question stands: how much of a danger does the current microplastic pollution level in the seafood we eat bring to the society? And how did the microplastic end up here in aquaculture? Let's move on to the next slide. Moving on, the objective of this research is to first investigate the level of microplastic pollution in commercially available seafoods in Selangor, Malaysia, and to identify its sources in the aquaculture industry. There are two methodologies that I use. The first one, primary source, by using gravimetric analysis. There are many methods to use to quantify and also to detect microplastics, such as using SEM, FTIR, or Raman spectroscopy. However, these requires significant amount of knowledge, background knowledge, in order to use them, and access that is quite limited. So therefore, I will be using gravimetric, which essentially uses mass in grams to quantify the amount of microplastics in these animals. And there are three steps involved. The first one is pre-experiment. Pre-experiment is important to ensure that there is no other microplastic contamination that could arrive, because microplastic has been detected to be detected in the air. So therefore, the air, the workplace has to be in a well-ventilated area. Sanitation with 70% ethanol and paper towels is important to remove any surface-level microplastics. Acid treatment of apparatuses is important to remove any residue from the apparatuses that is going to be used. Seafood sample collection essentially means finding the best seafood source that I could find, which in this case is from Hero Mart, which is the common digestive solution. 10% KOH preparation is essentially the digestive solution that is used to digest the matrix of the microorganisms of the animals, to ensure that only microplastics are present. And then the equal masses of animals are digested using equal 10% KOH inside orbital shaker incubator, and then it will be subjected to vacuum filtration where the change in mass of filtered paper indicates the mass of the microplastics. And then the reason why the mass of microplastics is there is related to secondary sources that I found, including finding reliable articles, journalism websites such as Voce and National Geographic, and books such as Google Books, Oxford IB Biology 2014 edition. Therefore, for my research, I have found it has the highest amount of microplastics at 1.0576 grams, followed by mussels, clams, shrimp, and squid. To put into perspective, 500 grams of cockles contains approximately a whole water bottle cap. And as we know that 1 gram of microplastic is found in 100 grams of cockles. So 500 grams of cockles is enough for us to consume a whole water bottle cap. And then the reason for this is because cockles, clams, and mussels are essentially bivalves, and bivalves have a specific type of water that is coming through them, like squid and shrimps, where they only eat smaller organisms. Therefore, plus cockles, clams, and mussels are eaten whole, unlike shrimp and squid, where we remove the internal organs, and also shrimp, we remove their heads. So therefore, the gastrointestinal tract is the most abundant in microplastics are not ingested. And you can see as well that the quantitative data also correlates with the qualitative data shown. You can see that cockles, clams, and mussels, the ones with the highest amount of microplastics, you can see that there is a consistent color, gray color area in the center of the filter paper, indicating that there is microplastics that is being trapped within the pores of the filter paper, unlike squid and shrimps, where they do not have that gray area in the center. The reason for this cockles, clams, mussel, the reason why all of them contains microplastics is because of the method of rearing. They are essentially put into cages and they are left to grow in the they put the animals inside cages, and then these cages are put in the seas, and after a period of time they would get them back. And the reason why this is the method, why this is the prevalent method of microplastic contamination is because the water is essentially showered with the main source of water pollution, which is fabric washing or laundry washing that the wastewater treatment centers, they do not filter these microplastics, and the clean water are essentially exposed to the ocean where it is the same habitat as these animals, therefore contributing to the microplastic contamination. Therefore, in conclusion, bivalves are more capable of ingesting microplastics compared to cephalopods such as squids and crustaceans such as shrimps due to their method of feeding. Exposure to polluted water primarily pollutes the rearing seafood, and however, in terms of its methods, it requires more focus, because the microplastic identification and quantification method requires standardization and more sophisticated apparatuses to improve data reliability. Further research is needed to evaluate quantity of microplastics that can invoke danger. There are some limitations in the experiment. For example, the methodology lacks identification methods to ensure that the microplastics are actual microplastics. It has an unreliable sampling method, and it has low reliability quantification method, a small sampling area, minimal types of seafood tested, and large filter paper pores. And the suggestion that I've given is by using spectropluorimetry quantification method, by using fluorometers to indicate the intensity of the indicators using NIRAD dye, smaller filter paper pores, and wider sources of seafood. And we need consistent filter paper condition. In theory, the challenges that I found is because of the laboratory and equipment unavailability. I required laboratory that is not within the areas of KMB. I would have to go to MGVI, which is the Malaysian Genome and Vaccine Institution, which is close to Agency Nuclear Malaysia. I would have to book a lab there, and I had to gain access to that industry where I have worked with other people who are actually investigating real world issues, and therefore that built my soft skills because I had that industry level experience and proper academic writing methods that are suggested by them. And other challenges include limited methodology because the methodology requires more sophisticated instruments due to the microscopic nature of microplastics. And also, obviously, in all research, we always have to have the necessary writing and multiple draftings. That is an issue that is faced by most of us. And I believe that in a way, these research have helped me to understand and to feel the experience of becoming a true researcher, where the skills that I've developed really helped me to understand the capabilities of what an industry like this can do. And that's all from me. Thank you for listening. I hope everyone has a great day. And if you have any questions, you can just contact me later on whenever you're free. And yeah, thank you for listening. Have a nice day.